Welcome, everyone. Thank you for joining us for today's track on Earth and human management of our microbial wealth. It's my pleasure to introduce to you Dr. John Thomas, Professor Emeritus at West Virginia University School of Medicine. Dr. Thomas is recognized as an international educator and global microbiologist, having lectured in more than 43 countries, as a clinical microbiologist in pathology, clinical professor in dentistry, and an adjunct professor in graduate school education for 54 years. His translational research emphasized biofilms and medical devices. Dr. Thomas has over 50 publications and multiple book chapters spanning his career. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Thomas's keynote presentation is titled Microbial Centric Aging, a Paradigm Shift. My name is Michelle Ashton, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers free continuing education credits. Please click on the Continuing Education's Credits tab on the top right-hand corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. John Thomas. I will now turn the presentation over to him.
Well, thank you very much, Michelle, and it's really my privilege and part of my psyche to want to continue to educate, even I think when some of us have decided to be retired and go on in the academic world, minus the actual classroom. But what I want to do today is go with you and hopefully create in your mind some of the enthusiasm that I'm developing with a topic of aging. It's somewhat topical to me, but it also clearly identifies an area where we have much to learn in the world and where we're beginning to learn that maybe the microbes are much more important than we've ever anticipated. In my lecturing capacity at West Virginia University, I developed a cadre of cadets and puppets. You'll see them listed here. My puppets help me create a theme. And so if you get a chance, one of the things I want you to do is to see the website, which is globalbugs.com, which under certain categories, in this case teaching, will describe the importance of these puppets and their relative themes to the lecture material that we're going with today. I won't use every one, I will use selected ones, but I want you to realize that this is all part of who we are. And the theme that I want you to address is, again, going to the website, something I began to think about, and that is we need to rethink microbiology. And I came up with an advertisement that sort of addresses that interest, and I called it Microbiology Reimagined. And on the website, there are actually 5 keys of what this new Microbiology Reimagined is. And one of them clearly is what we'll talk about today, is aging, and the concept that we need to deal about wellness. We need to recognize that there are multiple parts in who we are as far as our wellness, our earth, and the animals around us. And in all, I want you to carry the theme that says we live in a microbial world. This is the website specifically. You'll notice in the lower right-hand corner, I have the Center for Hologenomics and Clinical Studies, and that really addresses the theme of who we are relative to our microbial inhabitants, our microbiologic kin. And there's quite a bit of material here that helps supplement what I'm going to be talking about. So please do, at the moment you have some time, go to the slide, because it will have supplemental information that enhances, I hope, the lecture today. And as I already said to you, one of the key points is that, as addressed by 2 previous presidents of the American Society for Microbiology, We need to deal with one health, one wellness. And it addresses three things: the globe, animals, and the inhabitants, you and I, Homo sapiens. And it expands, if you look at the slide on the bottom, features that we need to consider when we address who we are, who contributes to who we are, and the wellness that each of those parts will play. If you think of it as a genetic scenario, and you look at how we interact gene-wise, there's often the discussion that the globe provides us through a variety of mechanisms, some information that is transferred and that ultimately impacts on Homo sapiens. It's slow. It takes millennium evolution. One of the thoughts I hope to generate in your mind is that if you address the impact of a genetic information provided by microbes, it could be fast. It could allow us to, in fact, not alter this theme of evolution, but clearly make it a theme which has an impact in our lifetimes. So keep that in mind. As we go through this, this is my first real thought on aging. And I'm going to address a definition because that's clearly something that's always part of a new topic. We need to be considering about who aging applies to, and the magical year of 2035, and I'll explain to you why as I go on. That slide is now. We in the United States, as a representative of the globe, are an aging nation. And in 2035, over 50% of our population will be 65 or over. That's incredibly important because, as you see in the globe, using some of the more recent Japanese data, we've always addressed on the left-hand side sort of the pyramid, which is changing in 205, and the forecast for 255 on a global perspective addresses another key feature here, that the female and male propensity to be equal in all things is not quite true. And this has an important point on longevity and aging, because look at the concept of who lives longer, and is there a reason for that which is genetic? And if so, can we alter it by using certain information-based genetics, which could be microbes? So think about that as we develop this theme. The idea of classifying aging and biologic aging as a disease has been around. It's not new, but it gained stature when people began to really give it some thought relative to how it impacts our costs and how it addresses research. So aging originally has been defined as a harmful abnormality of bodily structure and function, and it may have some specific causes, which I'll elaborate on a moment. But what are the benefits if we suddenly describe aging as a disease? We eliminate this sort of fatalism and we suddenly say there is something to be gained by recognizing its importance as a disease. And in the red at the last paragraph, it talks about, would we begin to address more gerioprotective drugs, regenerative medicine, precision medicine? Would we enhance grant support to address aging as a disease? And would we gain access to a better understanding of aging as a specific feature? It really began to facilitate world opinion in 2015 when in Brussels it was a major meeting and they said, look, it's time to classify biologic aging as a disease. In 2016, it gained more editorial support when they said we need to address the academic, pharmaceutical, and healthcare policies relative to pensions and funding perspectives. The U.S., again, a little bit slower, began to address this after it appeared in a meeting in Ghent where the NIH said, you know, there may be a real reason to address the international theme, and it's time to classify aging as a disease. And really interesting, a consequence of evolutionary neglect, not evolutionary intent. And there's a lot of information about that, and it has gained not acceptance, but notoriety, because there are those who clearly feel that this is not the way to go. And that's one of the interesting things about it, because this is a controversial subject. And those that opinioned had the opposite side refuted the arguments describing aging as inevitable. Aging cannot be avoided. And so there are very real opinions that have significant distinct thoughts. Those who agree with the theme that aging cannot be defined as a disease often address a diagram such as the one seen in front of you, which on the left-hand side merely lists some of those unclassified but yet important features of aging: loss of regenerative ability, loss of cell proliferation, in contrast to those that are clearly associated with disease on the right-hand side: cancer, arteriosclerosis, myocardial infarction. And so how do you relate to these two themes and yet still not just hinder the development of what I think is an incredibly important feature? And of course, as I'm going to explain to you in a moment, if you look at who we are and who is with us through evolution of Homo sapiens, where do the microbes fit? Are they the elephant in the room? Is there a missing link here to age and age-related disease, which could be shepherded by microbes? And Lustgartner in 2016 wrote a marvelous editorial that said, to ignore the genetic impact of our dual citizens is to miss the point. You cannot put them in a can and have them go away. So the bottom line is you'll see, my impression is that we need to readdress this feature, and we need to put it in a perspective that allows us to gain access to more data. And that's why we perhaps need to address it as a disease. Anyway, moving forward, let's go to section 3. Here I have 3 of my puppets. The one on the upper right-hand corner is one that deals with age. The other aspect is one that deals on the middle right-hand side with planktonic microbes. And then we've got Marco Polo, a game that I remember we all played. And I'll tell you why this is important, because we're going to talk about the evolution of the hologenomic theory. We're going to talk about, as I mentioned, our biologic kin, recognizing that microbes matter. And at the end of this section, begin to address, can we get more information and data relative to who the players are in this disease, perhaps, that we call aging, by the genetic tools that are now on the market. And I'm going to hopefully expand that theme as we address this whole idea of the hologenomic theory. The hologenomic theory has been around for a while, as I'll show in a moment in a slide, but in essence, in a diagram, it points out that the history of evolution involves both humans in the green on the left-hand side and microbes in the blue on the left-hand side. And they are often described as evolutionary paths, parallel but not integrated. The reality is, on the diagram to the right, that in fact we have evolved together. I like to call it intelligent symbiosis. They are our biologic kin. Our dual citizenship is passport directed for both those microbes and the human cells that make up who we are. This is perhaps better explained here because part of the hologenomic theory, as I'll again explain in a moment, addresses the host genome, the genetic information of our host, plus the microbial gene pool, both called as one the hologenome with the hologenomic theory. Certainly as we talk about the globe and what it means with animals and our partners in the globe, all contribute to the gene strength of Homo sapiens. But to avoid aging as a discussion with only discussions about the host genome and not the microbial genome or the micro-holobiont is probably incorrect. So if you see in the next slide, and I have Marco Polo, you remember as kids we used to play the game Marco Polo. There's 2 parts to that discussion. And basically the hologenomic theory brought to fruition and national attention at Woods Hole in 1989, said that, look, there's 2 parts to us. There's a dual partnership, and we're going to call us the Holobiont. There is a macro, which is the gene strength of Homo sapiens, and there's the micro, which is the gene strength of the microbial population that has evolved with us in parallel. And the holobiont, particularly in the microbial sense, has the option of doing three things. We can amplify what the microbes do. We can bring in novel strains from the environment. And we can have horizontal gene transfer, all addressing the micro holobiont, which is part of our dual citizenship and our biologic kin. Marco Polo. If you look at it in another perspective, look at the gene strength. And as I'm going to point out to you, in one case, we have 10 trillion cells with the macro holobiont, and we have over 100 trillion cells with the micro holobiont. And the Nobel Prize winner has described the gene as one of the three most important features of discovery in the 19th and 20th century. The bottom line is that we need to recognize these two work together, and that it's important that as an issue, they do have the feature, as shown by the movement here, that together we are a different, perhaps, perspective of who we are, recognizing that microbes matter. Very much, microbes matter. If you want to go and become very scientific on this point, we can do so by looking at the gene strength that we just described, the holobiont of the macro combination on the left and the holobiont of the micro environment on the right. And if you look at the numbers of genes that we have as far as Homo sapiens, about 23,000 approximately. These numbers vary, but approximately. If you look at the number of genes that are involved with our micro holobionts, the microbial next of kin, you've got about 316,000 genes, and that number continually evolves. And as I've said to my students, it's really, we, the human, we have in textbooks maybe one book of information genetically. The microbes have perhaps 316 more. That's the number that we talk about. And the reality is, if you take this as a theme, you can begin to address why it cannot be a feature of aging that does not include some concept of the impact of the genetic strength of our other equal partner who is and has evolved with us over time. That's the hologenomic theory. It really, in some ways, I've said to students all my life, it really is a anti-Koch, anti-Louis Pasteur theme, although both of those met with more pathogens and put the microbes at a rather relatively disadvantaged point. The bottom line is we needed to really play a more significant assessment of their benefit and not benefit to who we are. One of the thoughts then is, as this whole hologenomic theory has evolved, it's been supported by the new evolution of diagnostic procedures in the laboratory. And as you see in the center of the diagram is a tree, which sort of highlights phyla, but highlights the relationship of microbes as defined now by some of our new rapid DNA testing. And if you look at the outline on the left-hand side, it lists life in our taxonomy. We have over 5,000 to 10,000 species in Homo sapiens. We have a huge array of microbes in the genus, up to 2,000. And the bottom line is, how does a diagnostic laboratory, of which I was a director for years, begin to address this whole concept? So it's beginning to be apparent that there are really, in the taxonomy of the organisms that are part of our system, perhaps 10 that are critical, 6 are really important and become benchmarks of health and disease. And they are listed there for you: the Acinetobacteria, the Bacteroidetes, the Firmicutes, the Proteobacteria, the Cyanobacteria, and the Fusobacteria. Those are phyla. It gives us the chance of looking at ratios of not just 5,000 to 10,000 species, which would be incredibly difficult even with artificial intelligence. But it begins to give us a theme of who are the major groups that are within our system and how do we address them relative to health and disease in this new outline of the hologenomic theory. And one of the points that obviously we need to address is if you look at the organisms in the phyla, which I just listed for you in the diagram and slide before, and you look at sort of a schematic of age, you begin to get a visualization of how these phyla change. Now, certainly we could look at species within the phyla, and certainly on individual studies, could get a better handle on who the players are. But in getting the concept and beginning to measure changes of who our cohabitants are over time, this is the way that allows us, and one thing that allows us to perform. We can measure phyla, and we can measure them over time, and the new tools allow us to do so particularly rapidly. Which brings us to the point of DNA testing. A lot of the newness of DNA testing is, as I'll mention in a moment, related on ancestry. But in development of a test that looks at the micro-holobiont and the hologenomic theory, there are now several online procedures, test centers, which allow us to look at the microbial contribution so that we can get both the macro-holobiont and the micro-holobiont in a reasonable time frame and its implication in not only health, wellness, but disease. So the one that at the moment I have been using uses machine learning. It's the new Biome. It's not the only one. It's probably been the one on the market the longest. It uses advanced intelligence, statistical analysis, and it allows us to look at a sample, which obviously all of us have talked about, the second brain, if you will, or the combination of phyla within stool samples. And the offering at the moment is that you can do a single sample, or you can do a time lapse, which allows you to, over time, with changes in what you are doing, to gather not the DNA information of your host, that is the macro holobiont, but the micro holobiont. And it gives you, at this moment, a diversity score, a percentile of what phyla and organisms might be at proportion to those that have other characteristics similar to your own. They developed a diversity score, as I mentioned. I'm hopeful that we get more information as time goes on, but they have a learning center, as do most, and a list of publications which will give you, again, the concept that within multiple avenues now, we have the ability to measure the micro holobiont, which is part of the hologenome. Well, what about the macro holobiont, or the hologenome of our other partner? And obviously, there are a variety of tests online. Ancestry.com, 23andMe, new ones appear on Facebook invariably, daily. And all of them basically let us establish our family history. They allow us to link to a tree and maybe get some records. They're getting a little more sophisticated, establishing some health risks and traits. I have some concerns about this, but it's all part of the whole idea that if we can define aging as a disease and make us part of wellness, both from the microbial perspective and from our genetic host perspective, we will really begin to get a handle on how these two interface and how they are good and bad in aging particularly. More recently, as you probably also know, there's quite a number of tests appearing again on Facebook and other social media sites, those that will take your data and decipher it. And so you can send them your Ancestry.com or 23andMe or Nebula Genomics or whatever, and they will give you back information that is beyond what the scope of the initial testing was able to provide, or they will provide you information that is linked to not only your history, but some disease parameters. I'm going to caution you about this. We'll talk about this at the conclusion, but there's still so much to be learned. I think there is a rush to look at this data, but the fact is, if we make disease and aging a part of the scheme that is NIH recognized, is funded by research, particularly pharmaceutical houses, we will begin to have a means and a reason to look at these two combined together, the microholobiont and the macroholobiont. Well, having spent some time doing that, now that you know who you are, and now that we know why it's important to recognize two parts of yourself, and that this self is part of the globe, which can be influenced slowly through evolution or rapidly through time with microbial impact. Let's look at the theories of aging, and let's look at a way that I brought to fruition with my puppets to try to give students an idea of how important this whole idea of who we are relative to our cohabitants early in life. So first of all, what are the theories of aging? What is the idea of a microbial clock and a biologic clock? What are some of the common pathways in age-related diseases? If aging is a disease, how does age-related diseases play into this? And specifically, how do our gut-brain interface occur, again, developing the theme of the macroholobiont and the microholobiont? So let's take a look at that. I'm going to be using, again, some of my puppets, but the theme addresses what I mentioned early on, and that is that we have genetics, who we are. We have the globe, who provides animals, environment of who we are. And historically, most theories of aging address either a genetic component, an environmental component, or as listed by number 3, a combination of both. Genetic, environmental, and a combination of both. Rather simplistic, but nonetheless sort of easy for categorizing where things go. And only until recently did anybody begin to think about where does this category go with microbes? And that's what we're now beginning to address. So there are a huge reference, there is a lot of references and a lot of articles written about human aging and how cells and components of the cells particularly are affected by a variety of things. Some of them are listed here, just some. The literature is replete, textbooks are replete. You know, damage and loss of damage control, cross-linking within brain cells, build of amyloid between cells, failing in adaptive immunity, and the list goes on. Microbial DNA damage, nuclear DNA damage, buildup of senescent cells. All of those address what has historically focused on one aspect of our genetic strength, and that has been the macroholobiont. So the issue that I want to address, and that I hope to stimulate your interest in, is then where do the microbes fit in? If there is such a reality as a holobiont, and we have a hologenome, we can't ignore microbes. And how can they fit in? So as I began to address this topic of interest to me, about 10 years ago, I began to really think about this. I'm an avid runner. And when I run, I take a pad and I can jot down notes. It's a time when my mind begins to really function as a scientist as well as a runner. And it began to be apparent that, hey, we as humans can be classified sort of in 4 age groups. It's also easy to find data on these 4 age groups, and that we could look at young adolescents, the midlife, and then the aged person. And we could also find information about the costs because, in fact, as you see here written in red, 32% of all medical costs are spent in the last 2 years of life. If that's true, which it is, and if it's part of who we are, can we make a picture that will relate to the other side of this diagram, and that is who we are relative to a clock and our microbial composition as it changes with time. So what I envisioned to help, again, translate for audiences like you and the idea of teaching about the microbial clock is if you look at diseases, of which I chose 10, again, based on cost, and you divided them into a young green, which addressed those things listed there, autism, asthma, particularly mother and newborns, and we call this the infant microbiota. Who are the players, if you look at phyla that we've already addressed? Of those 6, which ones are key at this stage? The other side, if you look at adolescence and you look at diseases like diabetes and obesity, again, who are the players in the phyla? Who are the microbial micro-holobionts that have a feature involved in this? If you look at then adolescent, the orange part of the diagram, you can look at other parts of the disease process dealing with gut, particularly irritable bowel disease, gastrointestinal diseases, but what about vaginal diseases, osteoporosis? And then you look at, those diseases on an population which by definition is addressed as aged, is 55 statistically, but a lot of us have addressed this as 65 and over. What about the microbiota of an aged individual, particularly those that are in nursing homes, dementia, Alzheimer's disease? Who are the players in this environment? And my theme here was really also driven by a person I've had the experience of talking to and working very, very at a distance, is the Martin Blaser. Connecting multiple diseases through a single framework or underlying cause was his theme, and I've sort of taken that theme and applied it to the microbiota that we are part with in our Homo sapien relationship, and said, how can I show this in an environment which allows me to say to the audience, your microbiota changes with time, which we all know. It can be measured in time, and it's part of who we are. So if you look at the literature, and I started to do that with a great deal of enthusiasm, and you look at the concept of those 4 categories of green, yellow, orange, and red, and you look at the individual phyla which are involved, one can begin to look at diseases in those age groups, and with the newer molecular microbiology, begin to assess the prevalence of certain phyla, certain species that are found preeminently in those diseases. Now, is there a cause and effect? That's still to be proven. But the reality is we are a continual revolving cycle of microbes in our lifestyle. They can be measured now, and they certainly can be addressed in a particular disease process. What really made this more important in where we are going is not only then the microbes as part of our phyla, as part of our micro-holobiont, but what are those microbes in combination with other microbes at the particular disease and the age of the individual? What do they have in common as a function? What are these phyla at a particular age group doing functionality-wise? Do these microbes involve themselves in a particular capacity of protein metabolism, carbohydrate metabolism, energy metabolism, or lipid metabolism, just to begin to address a few? So not only can we begin to look at who's present at what disease state, in what proportion to the other phyla, we can begin to measure their function, that they at that age group with other microbes begin to alter or enhance. And does this help us in better clarifying disease state if we have a common pathway that Dr. Blaser so well described? Particularly now, as this lecture is addressing aging and the aged microbiota, what we need to do is begin to look at those aged diseases as, again, a potential clarification of disease, and look at them not only as what I just addressed, but functionality, but what about another key category, which is, what about those that we lose? Over time, as I'll show in a minute, we lose an awful lot and change a lot in this globe, and we change a lot about who our symbiotic organisms are. So that not only do we need to be thinking about what organisms are present, and what functions they provide relative to energy metabolism, and particularly gut location. But what ones are missing with time? Because if we can address missing as well as present, perhaps we have an alternative to therapy. And as we go through this, I hope you'll remember that there are 4 age groups that were sort of selected because they enhance the ability to educate, to describe, and to relate. But it's not that they're absolute, but they give you a clue. If we look at age and we look at microbes and we look at the revolving door with our globe and wellness, we begin to understand that, wow, this is perhaps an area of great research, but we need to have funding. The idea that I came forward with on the biologic clock some time ago was really enhanced by the Nobel Prize in 2017, because it looked at Homo sapiens, and it looked at a clock approach to who we are. And looking at way back in 84, but given, again, the award in 17, they found in fruit flies a period gene. And they further experiments showed that plants, animals, and humans adapt their biologic rhythms so that it's synchronized with Earth's revolution, day and night. If you will, a 12-hour clock. Not a 100-year clock in the microbial sense, if you consider us living a long time, but in a day clock. And they determined that it was attributable to a genetic encoded protein called the period protein, and that, as you can see, it accumulates in cells at night and then is degraded during the day. But it's a clock. And the idea was that this 24-hour clock included multiple functions, again, like what we talked about a moment ago, a common pathway for microbial interface with human, and a common pathway for human genomes, particularly on a synchronized 24-hour clock. And it says their result, for which, again, the Nobel Prize, they said the multicellular organisms, including humans, operate a 24-hour clock, which regulates functions including humans, us, behavior, hormone levels, sleep, metabolism, temperature metabolism, blood pressure, heart rate, and reaction time. It began, again, a theme. How do you do these things all at once? So I combined the clocks in this photograph, or in this PowerPoint, and you can see in the outside are those diseases that are attributable to often microbes that are in a green, yellow, orange, or red category. But you now must include the human, which is very much the hologenomic theory, because that's on a 24-hour clock, and the synchrony of the human clock, the biologic clock, with the microbial clock certainly gives us the ability now to begin to recognize aging as a perhaps disease that would allow us to look at synchrony between 2 clocks that are part of Homo sapiens. This has really been addressed in the most important area, and that is where we have the most microbes, as you are well aware, the gut. And is there a gut-brain axis of information transfer? Because if, in fact, this theme of 2 of us working together is to work, then there's got to be a means of communication. And as shown here, we have a gut-brain axis, which has been recognized, but it's very important to recognize 2 ways. The ability of the brain to influence the intestinal microbiota, and in the other side of that, on the right-hand side, the ability of the microbiota to influence brain and brain behavior. What is known to this point, if you kind of summarize all the points about this axis and about what it can do, the keys are, I've highlighted in red. It's bidirectional. It's clearly less than functional when we have gut dysbiosis or dysfunction of the microbes and phyla within the GI tract. There isn't a modulation from the brain of the gut microbiota. And bacterial products from the gut clearly can have activity through cytokines and gut hormones, which are, again, each of them elaborated in individual research, but you can get the idea from the slide here Then in looking at, again, in the red zone, aging, in dementia, we can look at the lower part of this diagram to the left, the dementia associated with microbes and their byproducts of metabolism, their functionality, and their influence through neuron transmission to the brain, as sort of, again, highlighted in the middle, addressing the actual schematics of how in certain dementia these issues can be further identified and studied. So you really need to take a step back and think about what we've already addressed to the hologenomic theory, the idea that we can, in time, measure our changes in a clock, be it a biologic clock, humans, 24 hours, or a microbial clock, lifetime, just chosen for 100 years. But then again, while running, I happen to think about, again, with my age and taxes and benefits of banking, I began to try to draw a comparison, a parallel. And what I want you to do is take a step back, think with me as I was addressing this issue, because banking has some terms which allow us to address a common path between banks and our microbial bank. We need to begin to recognize what is our portfolio, who are the players within that portfolio, who's our manager, your money manager in finances versus your microbial manager in your gut. And can you leverage, as is often talked about in banking, your microbiome? Can you leverage it to enhance disease management, recognizing, if you will, what we said a moment ago, functionality? And can you begin to really address who we are relative to the use of pre and probiotics, which are again a feature that I'm becoming very interested in, given their influence on the red quarter or red zone of our clock. So here's my theme. And I began to think about this, and you'll be surprised, I hope, how this is really parallel to, again, macro and micro. On the left-hand side are those things which I heard my banker and my money manager begin to say to me, and they begin to address on the left-hand side a financial portfolio. And we begin to invest early in life because we are looking towards retirement, which was all part of, in the center of this slide, my wealth. But as you begin to think about the vocabulary in the middle, your information manager, the fact that you wanted your portfolio in finances to have some diversity, you wanted to recognize gains and losses as time evolved, and you wanted to leverage what would help you as an advantage so that you could have wealth in your retirement and in your lifestyle. Now go to the right-hand side and you look at your microbial bank. The gut manager, as I'll explain to you in a moment, is a fungi, probably. At least a lot of information is pointing in that direction. Your new means of addressing your microbial micro-holobiont gives you the fact of measuring your holobiont. And you can measure it with time, particularly by recognizing what have you lost. And that's why the blue arrow is going down. Because does that address then a therapeutic intervention? Whereas on the left-hand side with finances, we talk about investment banking. What about nutrition banking? And what about its influence on age-associated diseases? Can you feed your microbiota selectively to address those organisms that might influence your dementia? And can you selectively influence organisms which have been lost with time, but maybe would help if you reestablish them as part of your microbial bank? That is your wealth. So I really developed this, and I think it's interesting. Again, it helps me teach. But if you look at the left-hand side, you look at the financial bank, we deal with, over time, income and stocks and bonds and retirement strategy. And it's all, again, recognizing that investment helps us maintain our wealth. And the wealth is the definition of our health. And we talk about gains and losses. If you look at the other bank, your microbial bank, it really has been influenced, as we saw by the clocks, by your normal flora, birth to death by where you live, your pets and animals. It's been influenced by your nutrition, by your diet, and predictably by your age. All of those make your gut manager incredibly important in response to the good and the bad of illness. And can you, in fact, alter that if you understand who your gut manager is and who is your manager of addressing who your microbial balanced features are? And I never thought about this, but I began to think about it again. And with a lot of the reading and in talking to investigators who are beginning to address the gut manager or your microbial manager of your portfolio, probably at least partly associated with Saccharomyces boulardii. Saccharomyces boulardii, if you read the literature, has a lot of good things and a lot of bad things that are being said about it. But the fact is, it is a yeast. It's generally, in a reasonably healthy person, non-pathogenic. It is obviously more often than not antibiotic resistance. And there are a number of published articles, particularly coming out of France, that describe the influence of a yeast in the management of our microbial portfolio. Really interesting information. So let's look at this a little bit further, because as we begin to look at what happens to us over life, and if you look at the left-hand lower ordinance looking at life, and we look at investments of numbers, and we follow the blue line up to retirement, we are generally in our financial planning always looking at that concept of numbers that we can live with, because your financial planner or your broker has established for you a limit of what you need to maintain your lifestyle. And those numbers are in the millions generally today with our issues. What happens when the market drops? What happens when you lose, as happened more recently, 800 points in a day? What happens when you lose 400 points in a day? And how fast do you recover in the financial situation? And this diagram is merely to point out that there are numbers and there are schemes and there are places that can address this, but how do you recover from this particular event, which we all have to deal with? I want you to translate that, though, and make it into a parallel. What about your microbial bank? What about your microbial portfolio? So again, we start with, if you look at the lower right-hand side, left-hand side, and you look at the lines that address mid-age, if you will, what would be our yellow quadrant on our microbial clock, and you follow it with time again, the blue line. We'd like to reach an aged microbiota which is compatible with life and health, not compatible with diseases, and not compatible with particularly in aging and age-related diseases. And if you look again at the numbers, and you look on the upper left-hand side, I've got listed bugs 1, 2, 3, and 4. If you begin to sample, what happens, as with uBiome or any other DNA microbial test, what happens when we use antibiotics? Either short-term, strep throat, if you want, wounds, if you want, or long-term diseases. What do the bugs, represented more often by phyla, begin to do as far as being eliminated? And how fast does it happen, or how often does it happen when these reoccur? Do they come back, or are they lost? So this diagram here begins to point out to you that now, again, the lower left-hand side, it's a loss of income. Your retirements and your stocks and bonds. And we talk about the number, about 10,000 people begin to say, whoa. If you look at the microbial side, the antibiotics interfering with our normal growth pattern, C. difficile. We could spend hours talking about the consequences of antibiotics and C. difficile, and we can talk about for hours about irritable bowel disease, because these clearly do address a consequence that, again, it's interesting to me, the numbers. If you lose about 10 to the 4 microbes, it's always associated with some consequence microbiologically, a collateral damage. And it's interesting that both of those, be it financial or microbial have sort of the same range. But I want you to recognize that one of the key features of either financial loss or microbial loss is loss of diversity. You will hear it said in the banking environment, diversity is key. You'll hear it from me and other microbiologists that health is associated with diversity. The less prevalent, but the more prevalent, but more frequent microorganisms are those that really establish who we are and what our health patterns are. So think about this as you address the next slide, which enhances what happens over time. And that is, you lose your microbiota as you age. You have an infant microbiota, you have an aged microbiota. And the key here is, can you acquire organisms to enhance or replace that? Or are you simply, as you age, never going to replace who you are on the micro holobiont? And that addresses the topic that I've spent probably the last 15 years on really looking at, and that is, can you leverage your microbiota? And in reality, we have developed a back to health scheme and a partners for life scheme, which enhance us by going to the references and allow us to say to you that whereas overall there are a lot of articles addressing health in a general sense, there are also a lot of articles addressing specific diseases. And if you begin to look at who we and how we have found the publications, at this website, we have begun to look at those that can be addressed by conditions, the probiotics, who the microbes are. There is an awful lot of information on the web that allows us to begin to categorize probiotics and prebiotics into those that are specifically targeted against a disease and they would help in back to health, or those that are focused on partners for life. This is a diagram of a PowerPoint of a lecture that I gave, but I want you to focus, if you look at all of these diagrams and colors within the human, represent out of 310 articles, those that are addressing topics. Obviously, the largest is the gut. And if you look at aging, we have reviewed 335. And the problem that I have with these articles in either back to health or addressing partners for life is that a lot of them are not of the quality that we need to be getting. So in looking at this website and looking at probiotics and prebiotics, what you need to realize is that we've looked at everything relative to the condition at the top of this bar, the study type, the strength, what kind of microbes they're looking at, and what probiotic was used. And the reality when we look at aging and our red quadrant, we recognize that there are publications that deal with theories and that those that deal with specific diseases. Unfortunately, probably half of what we measured and have looked at and have categorized for you on this website are not of a value that we would consider to be yet publishable, or at least if published, need to be verified by additional studies. And so you'll see the strength of the articles are listed from actually E through A, but they're there to help guide you to realize there's a lot that yet needs to be clarified. The reason I put the word international here is that probably 60 to 70% of the articles that are now in the literature are probably from sites outside of the United States. It does not in any way suggest they're inferior, but it does suggest a focus that's been addressed internationally first, and then in the US second. This slide is perhaps one of the most important because it also now, in looking at partners for life, begins to address that we can take a pill or a combination of phyla, or we can address a more natural approach to probiotics that are in fact in certain kinds of foods. And there's a lot being published about this. We simply don't have the time to go over it. But I want you to realize that there is now a natural selection that we may begin to choose how we feed the microbiota of our gut to address the bidirectional interface, which would have the capacity to either reestablish, rejuvenate certain microbes and reduce certain others that will help. And again, partnering for life, which is why the title was chosen. You can see here, we could feed selectively our microbiota through microbial-focused nutrition, addressing functionality, which is what we've already talked about. So these articles in aging lend themselves to the fact that aging has an impact and that can be addressed as a disease, and it has the ability to be altered by looking at the micro-holobiont. I did a sabbatical at Cardiff University in Wales, a marvelous time, and I had the opportunity to at least hear about people in Ireland. And again, a reference here is Psychobiotic Revolution, and it addresses where it was performed, and that is the book authors, the two listed here, and the fact that it's in Cork, Ireland, again, representing the theme that I want you to recognize that an awful lot of work is being done outside of the U.S. So in summary, in trying to put this all into perspective, I hope I have addressed your interest using puppets that you'll find much greater clarification on the website, but we'll address the theme of who we are, clocks helping us address a single pathway, both microbial and Homo sapiens, and that part of the recognition now is that in aging, we can begin to address who we are relative to options. And it reinforces the fact that our microbial partners, our biologic kin, are very important in our aging process. Now, you have to remember that I started out a theme that I want to keep in mind. We live in a microbial world. It is part of our wellness. It's part of the globe's wellness. It's part of animal wellness. I also want you to recognize as we go into this incredible future, there are some limitations. And when you read references and look at articles such as I described in a moment, recognize that as on the right-hand side of this diagram, we have an awful lot yet about the microbiota, obviously, that is still yet to be defined. And it will expand how this interface of clocks and aging comes into play. So is aging a disease? And is there a microbial contribution? I mean, everybody has to agree upon their own theme. What I hope that you will begin to recognize is that we clearly need a new definition. It does address a tradition of society and where we have been born and who is our lineage. But the reality is the new definition, fostering more research and money and the ability to gather the pharmaceutical impact into aging, is to not say, oh, it's just an age thing. We want to clearly address those things because there is a component that allows us to have some impact that heretofore hasn't been well understood. So again, the hologenomic theory, macro and micro, the idea of putting who we are together is that we really do have an impact. The microbes have an impact. And that the reality is in aging, my other theme I want to convey to you is that I believe we can age well longer through microbiology. Again, let me repeat that. We can age well longer through microbiology, but we have a lot to unmask. That's really it, folks. I have enjoyed this. I hope I've stimulated you, and I want to make sure you have the idea that there are references at my website. I have to thank my two financial managers and my CPA because they've gone with me and helped prepare this. But the last slide it highlights some of the well-known journals, or in this case, textbooks, that publications that I have used. Missing Microbes by Blazer, the Brain Maker by Dr. Perlmutter. The most recent book is called Cultured. These and the Psychobiotic Revolution all have information that will enhance your ability to understand the topic. Lastly, please recognize, again, my theme, we live in a microbial world, microbes matter. Don't trash your microbiota. Don't trash your mycobiota. because stewardship of microbes is what will allow us to live longer, better, and with a health that is enjoyable, not where we have been. I love these slides. I leave you with that point. Thank you very much, and I look forward to your comments.
Thank you, Dr. Thomas, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. As a final reminder, Dr. Thomas will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we'll see you next time. Goodbye.